Hello everyone. In the previous video, we learned about translation, and in this video, we'll be learning about the regulation of gene expression. So, what exactly is gene expression? Gene expression refers to the trait expression. So, a particular gene codes for a character or a particular trait. So, whenever the trait or the character is being expressed, that is our trait expression. So why do we have to regulate the gene expression? So we know that all our cells contain the same degree of genetic material. That means all the cells have the same DNA, have the same DNA. Now let us take two types of cell, our skin cells and our liver cells. Okay, so the liver cells, they produce bile, right? And our skin cells, they have melanin, they produce melanin, they have melanin, right? Now, let us say, for example, the liver cells, instead of producing bile, they start producing melanin, and the skin cells, instead of producing melanin, they produce bile juice. So you can see that this will create a dysfunction in the body. So, and this is not supposed to happen. So this actually does not happen in our bodies. The skin cells, they produce melanin, whereas the liver cells produce bile juice itself. So this happens in spite these cells have the gene expression which codes for both of these, and even the liver cells have the DNA that can code for both of these. But the only thing that is being expressed here for the liver cells is the genes for bile, and for skin cells, the genes for melanin. So you see, depending upon the function of our cells, the corresponding gene sequence is expressed. So that is why every cell has a different function, right? So that is why our genes need to be regulated. And to understand how the genes are regulated, we need to understand the term opera. So, opera is basically a group of genes that regulate other genes, that regulate other genes. And this regulation can happen at four levels in eukaryotes. It can happen at the transcriptional level, or during the post-transcription modifications, that is during splicing, or when the mRNA is being transported from the nucleus to the cytoplasm before translation, or during translation. So it can happen at four levels in eukaryotes. Since post-transcription modifications are absent in prokaryotes or bacteria, it can only happen at two levels in bacteria, that is the transcriptional level and during the translation. So translation can be the last stage where the gene regulation takes place, right? So now opera, as I told you, is a group of genes that regulate other genes. There are many examples for opera, like we have tryptophan opera, okay, or we have valine opera, operon, etc. So we are going to learn about lac operon, that is the lactose one. So here lac stands for lactose. So this lac operon, which actually is in prokaryotes, this is a kind of negative regulation. So let us see how it is a negative regulation and how it works. So first case is in the absence of inducer. What is inducer? Inducer here in this case is our lactose. And why do we call it inducer? We will see. So the lac operon consists of three parts. Okay, so first we have the regulatory gene, or we say the I gene, and then we have the operator gene, and we have our structural gene. So the regulatory gene, it codes for regulatory proteins which can either act as promoters or repressors. And then we have our operator gene. So we'll come to know the function of the gene later. And structural gene. So structural gene is basically that gene which codes for enzymes, etc. Now, when our inducer, when our lactose, see, the primary function here, what we are trying to achieve is whenever there is lactose present, we need to break down this lactose into glucose and galactose. Whenever there is no lactose present, we know there is no need to break down this lactose. So there is no need of the enzyme for breaking this lactose being produced. So whenever there is lactose, we need that enzyme which breaks down the lactose. Whenever the lactose is not present, we do not need the enzyme which the breaks down the lactose. So when the lactose is absent, we do not need the enzyme, right? So here in the structural gene, we have three other parts. We have the Z gene, we have the Y gene, and we have the A gene. So the Z gene, it basically codes for the enzyme beta galactosidase. So the beta galactosidase, what it does is it breaks down lactose into galactose and glucose. And the Y gene, it codes for permease. So permease, it makes the membrane of the bacteria lactose permeable to lactose. It makes sure that the lactose enters the cell. Now another enzyme here that the A gene codes for is the transacetylase. So this has the function of attaching a methyl group, not methyl, acetyl group to lactose. This is basically the function of the genes present in the structural gene. Now, when lactose is absent, we do not have to transcribe all these genes, right? We do not have to transcribe these into mRNA. We do not require that. So for that to happen, what happens is that, as usual, our RNA polymerase starts polymerizing as it reaches the code. So first it starts from the regulatory gene, and this regulatory gene, I told you, codes for a regulatory protein which can either be promoter or repressor. Since this is a negative regulation, it will code for a repressor. So uh, it forms an mRNA which is called the repressor mRNA, because this mRNA is going to help in the translation of the repressor protein. So this repressor protein is formed. Okay, this repressor protein which is there, this repressor it goes and binds to the operator region. So it binds to the operator region so that the RNA, while transcribing this, it will stop till here and it does not move further. Okay, so the RNA stops before the operator gene and it does not code the structural gene, so it stops till there. Now, in case our inducer was present, in case the lactose was present, the repressor gene, instead of going and binding to our operator region or operator gene, it goes and binds to the inducer, that is the lactose. Okay, and whenever this repressor gene binds to this lactose, it becomes inactive. So that is why we here have told it has become inactive. It becomes an inactive repressor. So in this case, the RNA polymerase is not blocked by anything here and it continues to make mRNA for these genes also. So then we get a lac mRNA. This lac mRNA will code for the following enzymes, and this enzyme, they go and they digest this lactose. They help in the breakdown of this lactose. So this is how this is a kind of like a negative uh, regulation going on here, because this repressor which is there, whenever the inducer is absent, it goes and binds to the operator so that the DNA cannot further be transcribed. But whenever the inducer is present, the repressor goes binds to the inducer and becomes inactive so that the DNA can further be transcribed to form the lac mRNA, which can produce the enzymes that break down this inducer, that is the lactose. So this is how gene regulation takes place. This is how lac operon operates.